P.Yadav in short, you can say. And I'm working as a principal scientist in one of the CSIR laboratory, that is Central Institute of Medicinal and Aromatic Plants, Lucknow. So our major focus area is to uh, work on the medicinal and aromatic plants, and we are working on the different aspects of the plants, like for the agrotechnologies, for the medicinal plants and also aromatic plants, for drug development part, for the chemistry part, for plant molecular biology. So in total, we are trying to, we have about the team of 100 scientists, and we are trying to develop the different technology based on the uh, medicinal and aromatic plants. So let's start. And here, my major focus will be that what are the different, approaches to develop the herbal drugs, and how we can develop the formulations using the plant drugs, and what are the regulatory aspects at present in India. So that we are going to discuss with this within this presentation. And if anyone have any query, he is most welcome or she is most welcome. So should I start, Dr. Megha?
Yes, sir, please.
Okay. Yeah, thank you. So I'm basically involved in two major activities as CSIR CIMAP. One is the development of herbal formulation, that is the prime responsibility that was given to me because I'm the person from the pharmaceutical background. So whatever leads come out from the institute, we need to develop into the different formulations so that the technology can be given to the different industry. And the another focus area of mine is the pharmacological evolution of the plant drugs, because we screen the different newer plants, different plant drugs, which has already have been reported uh, in the scientific literature. And they may be mentioned in the different our traditional drugs. So for that, I'm focusing on diabetes, wound healing, and psoriasis at present. So these are my two major focus areas at CMAP. Before going into the detailed discussion, I would like to give you the a brief idea that how we can proceed for the herbal drug development, and what are the very interesting examples which are the milestone in the area of plant drugs, because mostly we use the drugs, they are synthetic or the allopathic medicines for our routine purpose. Very few people use the plant drugs, and normally we have the feeling like not very important drugs are from the plant. So I will give you some examples. Actually, whatever drugs has come, basically they are they basically relate to the plant molecule, and out of those clues the modern medicine has been developed. So that we will take, like the very first molecule that was isolated from the compound and that was therapeutically effective. So very first pharmacologically effective molecule that was isolated from the opium poppy, that is a morphine. Okay. And opium poppy means Papaver somniferum, and that was done by a pharmacist. So as we know, morphine is such an important molecule that even till today, a number of painkillers are available in the market. But in the condition where no other drug works, so finally the morphine is given to relieve the pain. Like in the terminal cancer patients, the uh, where the pain is very, unbearable. In that condition, the morphine is used. So, this is not used in normal condition because, as we know, this causes the addiction. Okay, so that's why it is not very much used. But in the terminal patients, it is used as a, ultimate painkiller. And out of this morphine, many other drugs have been derivatized and they are being used. in today. And another important example I would like to give, the quinine, like malaria. Most of the Asian countries, they are suffering from the malaria. They're fighting still against the malaria, and a lot of casualties are there due to the dengue and the malaria. So the quinine drug was isolated from the Cinchona succirubra bark. Sorry. Yes. So the quinine was basically isolated from the cinchona bark. That is again a plant, and from quinine, and that was a very, wonderful molecule from quinine. Then we again modified the structure, and we came out with pramakine and chloroquine, other drugs which are still being used for the malaria. And yeah, artemisinin is another example for the malaria. Actually, many mosquitoes, they are they are resistant to chloroquine. So in that condition, particularly for the cerebral malaria, there was a need to develop a new drug. In about 1967, the in China, number of army personnel were dying due to the mosquito bite. So the government gave tasks to the scientists that we should come out with the some drug which can protect our army personnel from this uh, disease. So the Professor Yu Tu, which has been awarded finally in 2015 as Nobel Prize for the medicine. So her team took the clue from the traditional use of the medicine, traditional Chinese medicine. And from there, they isolated the compound artemisinin and gave to the uh, army personnel, and that was very effective drug against the cerebral malaria. So again, wherever there is a, need for a newer drug, most of the time we take the clue from the nature, and nature helps us to give some newer molecule or the lead to fight against these various diseases. Okay, so this is another example. uh, Before we go to the next example, I would like to mention here that, so actually, what is the approach, or how we can get the idea that from here we can develop the drug? So, friends, we have to keep in mind that, like India and China, these are the countries which have a very rich tradition. They have the traditional knowledge of like thousands of years old traditional knowledge with them. Like we have the Ayurvedic uh, knowledge with us. So we have a very long use of plant as a medicine and for various purposes. Similarly, in the China, they use the traditional Chinese medicine, the TCM. Okay, so they have very long use of plants for the various purposes. So these two countries, they are very rich in traditional knowledge. So that is why most of the time, India and also the China, uh, we try to take the lead, or we try to find out the clue in our traditional books. Like we have the Ayurveda, and also Chinese have their own books. So from those literature, they take the different clues and try to develop the drugs. But in the country like USA, which is not very old, so it is like 500 to 600 years old only. So that is why they don't have that traditional knowledge with them. So their approach is mostly to go through the high throughput screening. They have the technology, they have the facility, so they randomly take out thousands of extract and try to screen out for one particular uh, disease condition. So through this approach, the lead for the cancer was developed. The Wall and Warning group, they isolated the anti-cancer drug from the stem wood of the Chinese ornamental tree, Camptotheca acuminata, during the screening of thousands of plants in search of steroids. So actually they were searching for a steroid, but they finally came out with a drug which is effective against the cancer. And they also isolated the very popular and very effective drug in cancer, that is a Taxol, and now we call it the paclitaxel, and that was isolated from the Taxus brevifolia. So this high throughput screening, they could find out these two important drugs against the cancer. And so these are the two basic approaches which are used all over the globe for the drug development. Means either we can take the lead or the clue from our traditional knowledge, for our I mean traditional use, or we can go for the high throughput screening. And if we get some lead, and we can then we can optimize that lead into the form of a drug. So this is the conventional method of drug discovery, that like first we identify the what is our target. For example, if we want to develop the drug for the diabetes, so we have to we have identified that for what purpose we are going to develop the drug. So then we will find out that what are the different plant leads which can reduce the glucose level, or which can fight against the insulin resistance, which can increase the glucose uptake, cellular glucose uptake. So, first we identify that what is our target, for what purpose we want to come out with the drug, and accordingly we screen out our different extract or different pulp molecule for that purpose, and somehow or in some of the plant molecule, if we get the lead, then we also go for the preclinical studies and the small animal models. And when we get the activity in the small animal model also, then we try to optimize the lead. We try to uh, potentiate the lead, or we try to come out with a pure molecule which may be responsible for the activity. So in this way we optimize our lead, which can be more potent, can have the less toxic effect, and maybe more therapeutic value. So after optimizing our leads, we go for the again preclinical studies, that whatever we have fractionized or came out with the molecule, that is also effective in the animal model. And after doing all toxicity and efficacy studies, then we go for the clinical trial, phase 1, phase 2, and phase 3. And after these data, we go to the drug approval authority and try to approve the drug and market the drug. So this is the very conventional approach or conventional method of drug discovery anywhere that we adopt. Okay. Now, just so these were the some examples and the basic approach for the herbal drug development. Now we are going some definitions, because before we go that actually what we are handling, we should know how they are defined. Okay, and how the drug authority want that, in what form you should present the data, and in what form you should come out with the your plant lead. So normally we use herbal drug. That is the most common and popular name for the drug which are developed from the plants. And who are working in the field, they also use the term crude drug, the herbal drug preparations, herbal medicinal products, or herbal medicines, how they are different, and at what condition we use these different terms. So herbal drugs, they are mainly the raw drug, we can say, or the crude drug, another term that is used for the herbal drugs. So here we take any part of the plant, or I mean, we can take the whole or the fragmented or the cut plants or the parts of the plants, even the algae and fungi are lichen in an unprocessed state, and usually in the dried form, but sometimes fresh also. So basically, if we take out any plant material, that may be the leaf part, the fruit part, or the bark or the root, so that we will say it is a herbal drug. Like if we take the neem leaves, so the fresh leaves, we can call that, okay, it is a herbal drug or the crude drug. Then herbal drug preparation. When we take out any part of the plant to use as a drug, so we have to process it so that we can use it properly. Like if we take the any like leaf part, or like for a root part, like in ashwagandha, okay, so ashwagandha we use the root, so we take out the root, it will be called the herbal drug. But when we process the root, how we will process it? We will dry it in the shade. When it is dried, we will powder it. So this is the processing. So, or for the aromatic oils, we will take out the plant material. We will take the like aerial part of any plant, and we will go for the steam distillation. Okay. So through that, we will isolate the aromatic oil. Or we have any plant material, we can also prepare the extract, or we can express it to take out the oil of it, or we can fractionate it. So whenever we process it to get the extract to prepare the powder, it will be called the herbal drug preparations. Okay, this is the processed drug. And from when this processed drug, like we have the extract or we have the aromatic oil, after processing, we want to deliver it. So we cannot use directly as it. Although I mean, some of the Ayurvedic formulation, they allow it to use it as a powder. But powder is not very much, I mean, comfortable dosage form to swallow it and to take it. So that is why many pharmaceutical companies, they convert this powder into the tablet formulation or maybe the capsule formulation. So from these leads, when we prepare the finished products or any dosage form, so these are called actually the herbal medicines. So in brief, the raw drug will be called herbal drug or the crude drug. And when we process it, it will be called herbal drug preparation. And we prepare the final formulation out of it, that will be called herbal medicinal product or the herbal medicine. Okay, so these are the three basic, stages of herbal drugs. Now we will proceed further. I will give you one example that how we have gone through to develop the drug for psoriasis. What approach from starting we have taken up. So psoriasis is a skin disease. This is an autoimmune chronic inflammatory skin disorder, and whereas the hyperproliferation of keratinocytes and mesoinfiltration of the leukocytes and silvery scales, you can see here the silvery scales and plaques are developed over the skin surface. And mostly the inflammatory response is carried out by the cytokines and happens in the skin. Okay, so in 2013 we started the work on psoriasis with one of my PhD student, and before that we did not have any idea that how we should go for it, what are the different models for it, and how we should evaluate it. So that's why I'm discussing it, that most of you in the stage where you will have to select the research problem, then you have to deal with it, you have to find the ways how to go through it and how to come out with the lead. Okay, so as per WHO report, worldwide prevalence, the prevalence of psoriasis is about 2%, but in the developed countries, it is higher than this. It is about 4.6%. And that occurs in every age group of people. It doesn't matter in what age we are. So in India, more than 1 crore cases per year are for psoriasis. And what is the common treatment which is available? Methotrexate and cyclosporin. These are the immunosuppressant drugs. Okay, so they are used to treat the psoriasis because the psoriasis is a autoimmune disease. So this is the way that if we suppress the immunity up to some extent, this triggering of inflammatory cytokines on the skin may be controlled. So that is why the clinician, they give immunosuppressant, these two drugs in case of psoriasis. Other drugs are also there. And topically, to fight the symptomatic or to give the symptomatic relief from the psoriasis or the inflammation, they give the corticosteroids. These are the most common drugs which are used in case of psoriasis. Calciprotein, the psoralen, salicylic acid, coal tar, there are other drugs which are used in this condition. So when we started to work on psoriasis, we wanted to target the topical approach to treat the drug, not as immunosuppressants. So that was our primary target, that we want to come out with a drug which can be used topically, on the skin, and which can give relief, like corticosteroids give relief from the inflammatory condition. So these are the different problems which are, associated with the current psoriasis therapy. And when we found this, so what was our hypothesis? That we gone through the literature and try to find out the aromatic oils which have been reported as anti-inflammatory and antimicrobial or anti-cancer or antioxidant. So these were 3-4 major activity we tried to find out. And from literature, we find out some essential oil, they were giving good antimicrobial, anti-inflammatory and anti-cancer activity. And after that, we also try to find out if any mode of action is reported against those oils, that how they are reducing the inflammation in the skin or maybe some other places. Okay, so based on that, we selected 3 essential oil which were having good literature and having showing the good activity against the inflammation. Okay, so when we find out the drug with these antioxidant, anti-inflammatory, anti-cancer activity, so we screened these oils against the psoriasis like skin inflammatory condition. And for that purpose also, we saw in the literature that not very effective model was available at that time. So in 2009, we find out that Wonderfits, they reported imicomod-induced psoriasis like skin inflammation in mice, and that is mediated through IL-23-17 axis, and where they have more expression of IL-16, alpha, and IL-17, IL-22. And IL-17 and IL-22, they are very specific cytokines that are expressed in psoriatic condition, in human psoriatic condition. So that is why we selected this paper and tried to optimize the model in our lab, and we could optimize the model in about like 1 year. We could come out that how we should treat to the animals so that we can really get the significant difference in the normal animals and also the psoriatic mice. And we evaluated these animals based on the ear thickness, the PASI score. PASI score clinician use in case of psoriasis. Okay, so and in this process, we also developed one new method for scoring the skin condition through one skin analyzer, that is CosCam Skin Analyser, and we correlated this with the already available parameters. So this is the CosCam Skin Analyzer. This is simply the skin analyzer which is normally used in the cosmetic condition, because we were also working on the herbal formulation. So we had this instrument with us. We applied this instrument in case of to study the skin condition in case of psoriasis, and we could find out that yes, this is giving the very significant difference in case of normal skin and also the disease or toxin control group. So through, you can say this is the triangle we are getting in the disease condition, and this is the triangle we are getting the normal condition. How do we get it? Actually, we selected three parameters for the skin analysis: pigmentation, curvature, and the keratin content, and that changes in case of psoriasis. So through this, how they are increasing. So we were getting the higher area for this one. Okay, and also the different photographs which are showing. Okay, so these are the different conditions. So we also use that CosCam Skin Analyzer. And hello. Okay, so in normal condition, this is the PASI score, and disease condition, this is the PASI score we were getting. So through this, we could find out the significant difference in the disease and normal condition. Similarly, we also estimated the different cytokines which are expressed in case of psoriasis, mainly the TNF alpha, IL-1 beta, IL-17, and IL-22. Okay, so we use this model, and then we went for the skinning of different oils. Out of that, we got the lead as the lavender oil, which was reducing the this condition very significantly. So we can see that through cumulative PASI score, this is the score of normal animals, and this is the disease condition increasing very high. And again, this is the standard control where we were using the corticosteroids as a standard control, and this was reducing it very prominently, and also the lavender oil was reducing the PASI score very significantly. Okay, so these are the different evaluation we did for the in vivo, and then also we analyzed the TNF alpha and IL-1 beta. We evaluated the expression of these TNF alpha and IL-1 beta through ELISA, and also the IL-17 and IL-22. And we were getting very significant activity through lavender oil. Okay. So when we were, we got the activity in the oil, then the next question was, because the oil contains a number of plant molecule into it. So what may be the probable molecule which are responsible for the activity? Because until unless we define it, we cannot, come out with the proper lead, and which we can use as it were. Okay. So through gas chromatography analysis, the lalanol and lanyl acetate, they were the two major molecules in the lavender oil, and other molecules were also there. So we find out that lanolol oxide, lavandulol, lanolol, and the acetate, these are the major molecules. Okay, and in the literature, they have been reported for the anti-inflammatory, antioxidant, anti-cancer activity. Okay, and so we also go for the in silico studies of these molecules, that how is the binding energy to these receptors, and based on this in silico studies, we shortlisted the molecule lanolol and lanyl acetate. These were also the major molecules, and by using these two molecules, we first went for the in vitro analysis, where we performed the anti-inflammatory activity of these two molecules. So they were very potent. They were giving very potent anti-inflammatory activity. And after that, we also went for the in vivo studies and the safety studies. So, based on these study, we could come out with a good publication, and we also filed the patent out of it, and developed the product that is in the form of cream formulation. This is the publication that we have recently came out in May in the Journal of Ethnopharmacology. Okay. And this is the cream formulation that we have developed at CSIR CIMAP, and that has the for this product, the Indian patent also has been filed, and we are trying to further go for this formulation. So this was the, one approach that how we can, come out with any plant lead and develop the formulation also out of it. Okay. Now I will give a brief about how we can really make or develop the formulation part from the plant leads. Like my group initially developed one mosquito repellent cream formulation. So, and from the citronella oil. So our lead was in the form of oil, okay? And we had to make the topical formulation. So for this, emulsion is the best formulation where we can mix the oil and water together. For example, in the nature, milk is the best example of emulsion, where the oil like ghee is there and also the water part is there, and that has been mixed in such a wonderful way that we can dissolve the different drugs which may be water soluble or which may be oil soluble. Like in today's corona time, most of the time it is recommended to take the haldi milk, and haldi is not water soluble. So that is why we, and with the haldi or the turmeric, the major limitation is the solubility and the bioavailability, because it is not really, we are not able to make it bioavailable in the quantity which is required as a medicine. So that is why, we are using it, but not has come at a really a single drop. So with our traditional knowledge, we know that we can dissolve the drug in such a, natural formulation, that is milk, which will dissolve it and will make it more bioavailable. Okay, so this is the cream formulation. So we have the lead as a oil. So that is why we have taken the emulsion formulation, that is a cream, and where we use the different oil phase and the water phase, and the oil phase different oil excipients are mixed, and in the water phase different water soluble excipients are taken, we heat it and we make the formulation. I will give you one video also that how we can make it, and we prepared this one and we could license it to small industry, Metromed and Kirtiman group. Okay, so this is not only, one person who do all these things. So this is why I'm giving you an idea that formulation development part was done by me. The GC fingerprinting was done by one chemistry fellow, Dr. C.S. Chanotia, and parameditation was done because when we develop the cream, it should be safe. It should not be, induce any irritation and other things on the skin. So that was done by our veterinarian, Dr. D.U. Baunkale, and the mosquito repellent activity was done by Dr. Tripathi, who is an entomologist. So, there is a whole team which have to come together to come out with any plant lead or the formulation. Okay, and we later on we also did the different studies on it, and we could also come out with the publication uh, in the Biomed Research International. And I'm again give you just an idea that what is cream formulation. Like I was telling you, we take the water phase and the oil phase and mix it. If we simply mix it, it will get separated. Okay, but when we use the emulsifier with the oil and water phase, it balances both the things, and they make the, homogeneous mixture, not the solution, and we can get the stable cream or the emulsion formulation. Okay, so we have to use the different techniques, different emulsifier to prepare this formulation. This is one video that has been made in my lab, so that will give you an idea. Yeah. So in this way, we're taking the water phase and the oil phase. We can mix the excipients or the lead in these two different phases and can prepare the formulation. Here we, mix the aloe vera juice in the water phase, and we could develop or make the aloe vera cream formulation. And this is one approach how to develop the cream formulation. This is the HLV value based approach. Actually, first we try, based on our knowledge and literature, try to develop the cream formulation by trial and error method, by taking the different composition, different excipients. But sometimes actually we do not get the stable formulation. So in that case, uh, we determine the HLE value of the plant lead, and this is the method how to go for determination of the HLE value out of that, because with one of the oil we were not able to get the stable cream formulation. So we uh, took this approach, and based on this we could again develop the stable cream formulation. Okay, so this is another work we did in our lab, like Indian basil oil that is having very good antimicrobial activity, and from that oil again we developed the topical formulation, and which were showing the very nice antimicrobial activity, and which can be used as a antibacterial and antifungal formulation. And we also published this study in the Biomed International Journal. And in some cases, when we do not get the desired result in the conventional formulation, like cream formulation or the gel formulation, we sometimes we face the problems which are not solved through these formulation. So at that time we take out the help from the novel delivery based system, where we can overcome these problems. For example, the CIMAP, which is working on the plant extract or the plant oil, aromatic oil. So in one of the oil, the very good antimicrobial activity was there, but that was highly irritating. When we apply on the skin, it was causing irritation. We developed the cream out of it. Still, it was giving the irritation on the skin. So we could not use it as a, cream formulation. So for that purpose, this was the oil actually, Mentha spaghetti oil. And so in that case, we prepare the Microsphere formulation out of these oil, which helps to release the oil in a, controlled manner. So very less quantity of the oil comes on the skin, and slowly it get released. So that is why it was not causing that much high, irritation on the skin. So out of it, we did the different characterization parameter for the microsphere we prepared, and we did the skin irritation study, and we could see that the cream formulation which was made by using the microsphere of the that oil, it was not giving the irritation. Okay, so for such condition we can develop the different sustained leach preparation or other preparation, so that we can, do not get the side effect of the drug. Okay. And this is will give you an idea how to make the gel formulation. Like again, we have used the aloe vera gel, and aloe vera is most commonly used in the form of gel. We use on the skin because it is having very good wound healing and the moisturizing effect on the skin. So how to develop the gel, or how to prepare the gel? So this is actually an aqueous formulation where we take the water and mix into it the different excipients, and the important excipients are the polymer, which increases, or which enhances the viscosity of the uh, water. Okay. And then we add the gelling agent like triethanolamine, which actually changes the pH of the medium, and which helps polymer to come into the form of a matrix. Okay. So this is the basic approach. Yeah. So these are the few activities that I have shown, that is, we practice in our lab. and try to tailor the different formulations. So friends, now coming to the drug regulatory aspects, because now I think very less time is left. So in very brief, we will discuss that what are the different, approaches to approve the drug we develop or the formulation we develop, and what are the different agencies that give approval, okay, uh, for herbal drugs. So actually, what term we use mostly for the plant drugs like herbal drugs, if we go to the regulator and we say that we want to approve this herbal drug, so actually the drug authority does not approve or have any term like herbal drugs. So how they have defined actually these different leads. So let's see. First, we should look into that what are the approaches in the, other parts in very brief, I would say. Like in Europe, I have already told like the countries which have the traditional knowledge with them, so they have the different approach. When does not have the traditional knowledge with them, they are also having the different approach. So in Europe, like they are also using the plant uh, for very long. So the very first lead or the approval they gave with the traditional medicine uh, use provisions accepted on the basis of the sufficient safety data and plausible efficacy, which they are being used for long ago. Okay, and so based on their traditional knowledge, with their based on their safety data and efficacy, they approve the drug uh, as a traditional medicine. And another approach they use, that well-established medicinal use uh, of the drug that has demonstrated the scientific literature established and the active substance of the medicinal products. And uh, I mean, with the traditional use, if there is published literature, very validated scientific literature is available. So based on that, they also recognize, and based on the traditional use for the 10 years and also the efficacy and safety data that have been produced scientifically. So this is another category of the drug they approve. And the third one is if they do not have even any traditional use, and if any company want to come out with altogether with a new drug, and, with its own R&D approach or the drug development part, so they have to develop or they have to generate all the data. They have to go through all the studies, means like all preclinical studies, in vitro, in vivo studies, the lead optimization part, and again the clinical trial, then only they will approve this drug. Okay, so these are the basic approach in the Europe. And in US, they I mean they have given the guidance for this, the guidance to industry for the botanical drugs, that how you can approve your drug. So it may be like because they don't have the very rich traditional application of the plant drugs, so they take almost all the drug. If you want to approve your plant seed as a drug, so they took it as a new drug application, and for that you have to submit the IND, that is a investigation new drug. What is the meaning of this? Actually, whenever we develop the drug, if we have completed the preclinical efficacy uh, of the drug, we have the toxicity data, we have the quality control data. Okay, so with all these validated data, we go to the drug authority, okay, with all the data, and we file the application that is called the investigational new drug application. Okay, so this is before clinical trial, and this we file for carrying out the clinical trial of the drug. So before a clinical trial, This is called investigational new drug. Okay, when we go for the clinical trial and we have come out with a positive result of that out of it, like we have gone through phase 1, phase 2 and phase 3 clinical trial, and we found our drug effective and also safe. Okay, then after that we will file the new drug application. Means finally we give application to approve our lead uh, as a new drug. So that will be new drug application after completing preclinical as well as the clinical trials. Okay, so US FDA, they take all the drug as a, I mean, a new drug investigational new drugs application and all, and then they give the uh, drug approval after the clinical trial. Okay, they do not consider the traditional knowledge. You will have to generate all the data which is required for any drug approval. Okay, uh, if we have the some traditional use, then in some of the data they can give relaxation, but still you will have to generate the data. Like US FDA has issued a marketing authorization to a topical cream containing standardized green tree extract as a US botanical for treating genital warts after evaluating the respect to investigation new drug application scientifically. So this is, I mean, very different application of this extract. So they had to standardize the green tree extract, and then they had to go through the preclinical and the clinical evaluation, and based on that they approved this drug, a topical cream for the uh, treating the genital warts. Okay, in India actually we have the approach, like already we have Ayurveda with us. So various plant drugs were already in use for say thousands of years. So based on that use and the safety which is already proved in humans, the uh, drug authority, that is the Ayush Mantralaya, gave approval for these type of drugs. So the classical ASU drugs, okay, that is Ayurvedic, Siddha and Unani drugs, which have been mentioned in the classical books of Ayurveda or Siddha or Unani. They have given the name of about 54 books that the drug should be mentioned in any of these. And if you are making the same formulation, you don't have to generate any data. You can go directly for the marketing. Okay. But if you are, I mean, for example, if we are preparing the um, Avle formulation like Chawanprash, and we are making exactly as mentioned in the Ayurveda, so we don't have to take any approval from the drug authority. We have to just register our company and we can go for the marketing of it. But if somehow we are deviating from in any way from that lead or from the formulation, like haldi that has been mentioned in Ayurveda very well for the different purposes. But if we are going to prepare the cream formulation from haldi and that is not mentioned in Ayurveda, so we are changing the formulation. So these type of formulation uh, are called the patent or the proprietary medicines because we are changing the dosage form. Okay, so in that case we have to generate some data, we have to prove the efficacy, we have to prove the safety of it, and we can market this drug as a proprietary medicine. And but the lead will be again from the uh, Ayurveda, so it will be considered as a safe. Okay, so yeah, in India, the Ayurvedic Siddha and Union drugs have been under the purview of Department of Ayush, and they approved and marked the drugs. Yeah, but if any drug which is not been mentioned in Ayurveda, then how they will approve it? Then we will have to go through different, drug authority. In this case, CDSCO, that is called Central Drug Standard Control Organization, where the Drug Controller General of India is the highest authority. Like we are, we can see everywhere nowadays for vaccine approval, DCGI, the Drug Controller General of India. The name comes many times. So this is the ultimate drug authority which gives approval for the drugs other than the Ayurveda, Siddha or Unani medicine. So other than these drugs, they give approval. So like for the ginkgo biloba tablets, okay, ginkgo biloba, these are not Indian drugs, and they prepared the tablets. So this was considered as a new drug. So that's why the DCGI or the CDSCO gave the approval for this. Again, the Silimarin capsule. So this was the foreign plant, Silimarin. So that is why the DCGI gave the approval for this. Okay. So for Ayurvedic drugs, Ayush department gave approval. For other drugs, the uh, CDCSO or DCGI gave approval. Okay. But now in between, there was a challenge that the plants which are not mentioned in Ayurveda but are available today in India, if we develop the drug from those plants, then how should we go for approval of those plants or those drugs? That was the big caution, and many companies faced the problem for this, because when they were going for to the Ayush department, they were saying no, it is not mentioned in Ayurveda, so we cannot give you approval. And when they were going for the uh, DCGI, so they told yes, these are not the drugs which has been properly like the new drug, they have been generate the data, then we cannot give approval. So there was a caution, if the drug which are developed from the plants and the plants are not mentioned in Ayurveda, then how should we go? So that is why a new drug category in India has come, and that has been forced from 2016, that is called phytopharmaceutical drugs. So this drug category, which is not Ayurvedic, which is not the synthetic drugs, but the plant drugs, but not mentioned in the Ayurveda. So these are called phytopharmaceutical drugs, and the approval of these type of drug or the drug category we will get from the DCGI, that is Drug Control General of India, and the organization is CDSCO. Okay, so they have come out with the notification and different regulation they have given, and they have given the condition that what drugs will come under this category. So in very brief, I would let you know that phytopharmaceutical drug includes purified and standardized fraction with defined minimum 4 bioactive. Whenever we prepare the extract, it has many molecules into it. And out of this, at least we have to identify 4 phytomolecule. And we should be able to um quantify these 4 molecules in the extract which you are or we are proposing as a drug. So this is the minimum requirement. Okay. So minimum 4 bioactive or phytochemical compounds qualitatively and also the quantitatively, means we should be able to find out the quantity in the defined uh, drug. Okay, of an extract of a medicinal plant or its parts for internal or external use of human beings or animals for diagnosis, treatment, or mitigation and prevention of any disease or disorder. But this drug category does not include the administration by the parenteral route. So you cannot, develop the drug for parenteral use. Other than use like topical use, like systemic use, we can develop the different formulations. So this is the new category, and the CSIR is also working, DBT, ICMR, different organizations are working to develop the drug through this category. And these are the different, detail regulation that how we can define it. I'm not going through the detail of it. And what data has to be generated for this. So this is a very detailed list one has to go if we want to approve a drug as a phytopharmaceutical drug. Okay, and different stability and safety, in vitro, in vivo, then again clinical. I mean, like the investigation new drug IND filing, we have to go for it, and then for the NDA. Okay, so these are different data has to be generated. So that's all from my side. Recently in 2019, friends, I got the ICIMAP prize from our science minister, Dr. Harshvardhan, for different drug development part. And when I was in 2016 in Boston, USA, with Professor Tochlin, who is a known figure for the nanomedicine, who has developed a different liposome and micellar formulation of the drugs. And when I was coming back, and this is our team. So all this work, what I have presented here, is the actual efforts of my students who have worked in different capacity as a project assistant, PhD scholar, and the postdocs. Thank you very much.
Thank you, sir. So students, now we have time for question and discussion. So you are free to ask.
Hello, sir.
Hello.
Good morning, sir.
Good morning.
Sir, apart from the formulations of aloe vera cream, so
Yeah.
sir, I want to know, sir, you told us aqueous phase and oil phase. So I
Yes.
want to know, sir, in which phase is the continuously phase, sir, for aloe vera cream formulation, sir?
Which phase is the?
Continuously phase, sir, because sir, you
Yeah.
put some oil in the water, aqueous phase. So that's why I want, sir, just.
Yeah. Water is the continuous phase in this condition.
Okay, water phase, sir.
Yeah.
Okay,
Yeah.
thank
Okay.
you, sir. And second
Yeah.
question, sir, apart from the tinctures and extracts, sir, so in both, sir, we take active constituent. So, sir, what is the difference between the tinctures and extract process, sir? Because, sir, we take active constituent, no, sir, from the medicinal plant.
Okay.
So, sir, what is the, sir, difference, sir, tinctures and the extract process?
Tinctures and extracts.
Yes, sir.
Actually, tinctures, they are the alcoholic extract of any plant material, okay, where the solvent is alcohol. But in the extract, you can take other than alcohol, like other organic solvents, ethyl acetate you can take, or you can take like, the chloroform, not chloroform, hexane extract you can take, or the petroleum ether you can take. So if you take other organic solvents, in that case, that will be, extract, or you can even prepare the water extract.
Okay, sir.
Okay.
And sir, my last question, sir.
Yeah.
Actually, sir, I am pursuing the MSc in
Okay.
CSIR NISCARE New Delhi, sir.
Okay.
And my discipline is the Science Communication and Information Resources.
Okay.
So, sir, after the completion of the MSc, so I can join and apply in the CSIR CIMAP, sir. Also, I am eligible.
For what position you want to apply?
As a PhD, sir.
For pursuing PhD.
Yes,
Okay.
sir. Yes, sir.
Yes, you can apply at CSIR CMAP, but for that you will have to qualify the CSIR net examination. Okay, from like life sciences. Okay, when you, because we need any fellowship through PhD you can go through, है ना? So CSIR net exam is the minimum qualification for that.
CSIR net. So also, sir, UGC net, sir, is eligible.
Yes, yes, and even the DBT, they also conduct the GRF examination. So you must have any fellowship program qualified.
Okay, sir. Actually, sir, I have done the B Form, Bachelor in Pharmacy.
Okay.
After that, sir, I am pursuing the MSc in CSI NISCARE, sir. So further, sir, planning, sir, I apply in the PhD in also CSIR, CDRI Lucknow, and also CIMAP, sir.
Okay. Yeah, you're most welcome.
Okay. Thank you, sir. Thanks a lot, sir.
Okay.
Good afternoon, sir.
Ji, good afternoon. Ji.
Sir, I want to ask one thing. As you mentioned in the lecture that turmeric has a low bioavailability.
Yes.
And can't be used as an independent drug. So, can't we enhance the bioavailability of the curcumin by preparation of lyophilized liposomal curcumin nanoformulation?
Yes, I mean, the efforts has been made through this also. And if you, put a keyword in the Google or PubMed, the curcumin or the curcumin based liposomal formulation, you will get a number, hundreds of papers. But people are trying, but with, I think till now we have not come out with the formulation which is having, which is showing the desirable viability, and we can, give. We can use as a drug. Means with drug it is not only the one thing, we should be able, it should be stable also, it should be not very costly also. So some or other way we are still struggling with the formulation that we can come out. So that is why the most conventional formulation of the turmeric is used, that is tablet formulation. So out of that, whatever quantity is coming in our body, the people take it, and it's very popular in Western world.
Okay, thank you, sir.
Okay.
Good afternoon, sir.
Good afternoon, Nitish.
Sir, I have a doubt regarding formulation, sir. Sir, I'm doing a project related to balloon veins, an it's a immune modulator, sir. Immune stimulator. Immune stimulator.
Okay.
Balloon vein, cardio serum, spermum halocum. In Tamil, it's Tudwal. It's so, sir.
Yes.
Hello.
Hello. Yeah, I'm able to listen you.
Say it's in English. It's called as balloon vein.
Okay.
Cardio. I using it for as immune stimulant, sir. To prepare a tablet of that. It's 100% herbal tablet, sir. For anti-psoriatic, I have used turmeric and garlic extract, sir.
Okay.
I got a problem in its moisture content, sir. It is too hygroscopic in nature, sir. To overcome that, I used a cornstarch, but it was not good in that action, sir. Can you suggest me some other excipients to overcome this, sir? This hygroscopic nature?
Yeah, but I'm not really aware about the natural excipients which can help you in this, to overcome this. But definitely in the pharmaceutical excipients, because of, you can see a number of drug preparation, like from the Himalaya. Okay. Like tulsi tablets, they are having, like haldi tablet, they are having, the neem tablet, they are having, but they are using the the pharmaceutical excipients into their preparation, because when we go to use the plant material as a excipients, a number of different problems are there. Okay, that it is difficult to overcome out of this. So that is why I think I would advise you to use your plant lead, but use the other pharmaceutical excipients which will give you a elegant formulation. You can use like lactose, you can use as an excipient to give it the bulk, which so you will be able to punch it and make it the formulation.
Yes, sir. Thank you, sir.
Okay. Yeah.
Hello.
Yeah.
Sir, I am Swapnil from Maharashtra. Sir, my question is general. My question is general. There are internship facilities are available in their CSR institute, like aromatic institutes or something.
internship
Yes,
available?
Swapnil. Yeah. Internship, I mean, during masters, we give facility to the students that they can do their dissertation at CSIR labs. Okay. So always, I mean, at different time, you can go through the website of wherever you want to pursue your dissertation or something like this. So there is a call, so you can apply, and we, with some fees, they allow you to work for like for 6 months or sometimes for 1 year, where you can do your dissertation and also learn various techniques.
Thank you, sir.
Okay.
Good afternoon, sir.
Yeah, good afternoon.
Sir, I have filled that form which you have said about internship and dissertation work in MSc. And sir, but
Okay.
but how we can know that we are selected for that or not?
Definitely, you will receive an email. If you don't receive an email, then again you email them after some time. You can email to HRD. If you don't get any answer, you can email to even director. So director definitely will forward it to the concerned authority, and you will definitely get the answer for that.
Okay. Okay.
Yeah. So I think Dr. Megha,
Yes.
yeah.
I guess that I have no more questions. And if you have
Yeah.
any, mail me, students, or you can drop down the message in WhatsApp group. So I'll direct it to Dr. P.Yadav. So it was really very informative, sir. And here I want to say that we are in Srishti, we have herbal products for human as well as some agriculture products, and there are some veterinary products too.
Okay.
But our formulation, we are facing the problem of mixing the oil and extract in
Okay.
product, and
Okay.
human formulation also, after some time the oil is separated from the cream. So that's
Yes.
the problem we are facing. We have tried many emulsifier, but
Okay.
I think the lab team is not able to solve this. Yeah.
Mm-hmm.
So I think I'll talk with Professor Gupta, and
Okay.
we can go officially for this collaborative work.
Okay, I mean, that will be really wonderful. You're most welcome, Dr. Megha.
Yeah, thank you so much, sir. And I have also learned so many things because I am also a part of Srishti Lab, and we are trying for this formulation. And those herbal formulations are prepared from grassroots practices that
Okay.
we have a huge database, as you know.
Yeah.
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 So, definitely, sir, we look forward for your support in such workshop. And
Sure.
at this age and this stage of education level of this undergraduate student, they are getting the information from scientists like you. So it's
Yeah.
wonderful. Thank you so much for your time, sir.
Thank you, Dr. Megha. This is really a very nice and very wonderful program because this gives a feel of how the research we can do, and they get motivated also, and they have an idea and what we are doing in our different labs. So really congratulations to you and your team. Thank you very much.